Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs, Executive Director of the Finding Genius Foundation and host of the Finding Genius Podcast. My guest today is Monica Medina Munoz. She's a professor of biology at Penn State. She's also a principal investigator. We're going to talk about ecology and evolution of marine organisms. So, Monica, thanks for coming.
Oh, thank you for the invitation.
Yeah. So what are you studying in your lab? You have your own lab, after your own namesake. What's the focus of the research there?
So we studied several things, but I guess the main thread is symbiosis between cnidarian hosts. Cnidarians are animals, marine animals, and their symbionts, microbial symbionts, both algal symbionts and bacterial symbionts, and the interactions between them.
What's a cnidaria? Is it like a snail or something, or what is it?
No, cnidaria is a phylum of marine animals, some of the first animal phyla to evolve, and it contains jellyfish, corals, anemones, hydra.
Okay, I can picture them now. Huh. So they're symbionts, these bacteria and everything, are they symbionts that leave their body and hang out in the water around them and then go back in, as in some squid with bioluminescent bacteria, or are they inside them the whole time that they exist?
So there is one symbiosis in particular that is fairly well known, and that's between the cnidarian host and Dinoflagellate algae. These are unicellular protists that are photosynthetic, and they're endosymbiotic, so which means they live inside the host tissue, and they capture sunlight, and then they translocate photosynthetes into the animal host, and the host in return confers protection and inorganic nutrients and a place where they can do photosynthesis safely. So this is an important symbiosis that has been studied for decades because it's the essence of coral reefs. The interaction between these dinoflagellates and corals is what gives the coral the energy to build the skeleton that ultimately makes the coral reef three-dimensional structure where all the other creatures live. So that's a very well-known interaction, and the dinoflagellates can be free-living as well as endosymbiotic. The host, on the other hand, can live without the algal photosymbiont for a very long time, and that's what is known as coral bleaching. And climate change is having a severe impact on these symbiosis because when it gets too hot, the symbiosis breaks down, and then the algae leave the coral host, and the host doesn't have enough energy to survive, and more corals are dying from that. So it is a facultative endosymbiosis from the symbiont perspective, but it's more of an obligate symbiosis from the host perspective. So
where do these dinoflagellates take up residence? Like literally in the cracks and crevices of a coral, or like where do they go? Into the cells, in between the cells?
Yeah, inside cells, inside the tissue of the animal host. So they literally go inside. Inside, yeah, cellular processes have to take place, like phagocytosis. The symbiont is engulfed into a vacuole, and then the vacuole is transported inside the cell, and then it forms what is called as a symbiosome, which is a specific vacuole to contain the algae, and then the host does not digest the algal cell, but it maintains it. as an extra organelle.
And has this been observed to happen, like in a lab, in a dish, or like, when does this happen?
Well, it happens. So the symbiosis tends to be established early in the life cycle of a coral or a cnidarian. In the jellies, it's also the case. It's a little different. I can tell you about the jellyfish in a second. But in the coral, they have they have sexual reproduction through coral spawning. So they release all their gametes into the water column, and they fertilize while they're floating, and then they develop floating in the water, and slowly they become a swimming larva, and then the larva starts to find a place to settle. And in this process is when they develop a mouth, and when they have a mouth, they can start eating stuff. And one of the things that they ingest is the dinoflagellate, and that's in the ontogeny of the host. That's when the symbiosis gets established, and then the rest of their life they live within the coral host unless there is thermal stress, and then they leave.
So has anyone observed from the time that a dinoflagellate is eaten the whole path it takes to get to its final resting place inside of a cell?
Yeah, there is. This has been observed through microscopy. TEM and SEM images have and confocal imaging. There is a lot of high resolution microscopes that you can follow the sequence of infection in the early life stages of the animal.
Well, does it look like the cells, the host cells, are they actively going out and are they treating it as an infection, or are they deliberately engulfing them? And I mean, does it look like an immune response, this endosymbiosis, or what does it look like?
Yeah. To some extent, with gene expression analysis, we did some of that early on in the microarray era, and the microarrays technology didn't have the resolution that now things like RNA-seq have. So when you look at gene expression through transcriptome analysis, people are detecting some sort of immune response of recognition of cellular recognition of the algal photosymbiont.
Oh, but okay, so it looks like there is recognition, and therefore it's a phagocytosis, or it's like an endocytosis?
It's like a phagocytosis.
Okay, so what's doing the phagocytosing? Is it like immune cells from the cnidaria that are doing it, or other kinds of cells?
In the gastrodermal cell, in the gastrodermis, they are engulfing the particles, that they eat. So initially, they're not, I don't think they're actively seeking out, like the larva is not chasing after the photosymbionts. They kind of bump into them, and then they engulf them, and then the winnowing process kind of takes place where they trigger this recognition cascade, and they have preferences. There's many species of algae that they can engulf early on, and this is also, we've observed this in corals, and we've also observed it in jellyfish. The jellyfish actually uptake them not during the larval stage, but during the polyp stage. So that's a little different. But it's also when they have a mouth that they start taking them up, and then they don't take up all species of dinoflagellates. These photosymbionts are within a family called the Symbiodinaceae. They used to be a single genus, and now there are multiple genera within a family because the diversity was so large that it was necessary to do this taxonomic splitting into multiple genera. So now we understand a little better, because now we know there are many, many different species, and there is pairs that have co-evolved, but the babies have a lot more tolerance for uptake of different species. Some of them they flat out reject and they never take them up, and some of them they take up until a better one comes along. So people have looked at sort of the photosymbiont diversity over different stages of development, and you see that in the early months of colony formation after larval settlement in corals, or even in jellies. In the jellyfish, the infection triggers a developmental cascade. So the the polyp metamorphoses into a baby medusa called a phyra. So this process can be triggered, but there is already a disconnect if it's not the right one. So there is very specific recognition mechanisms. What they are in terms of each species recognition is poorly known or not known really, because we're only starting to examine multiple host algal combinations and in corals and in other cnidarian species. But we know that there is specificity.
Do these dinoflagellates stay inside the cells forever through the life of the organism? And are they passed down? Do they ever come out of the cells? Or they just stick with the cell until both die together?
I think it's a range of options. It depends on the host species that they have. Some species are spawners, so that means I described them before. They release the gametes into the water column, and the gametes are not infected. So every generation they have to acquire the algae from the water column. But there are species, I think at least I reported one or two, where the egg is seeded with the symbionts. So the mother colony passes its photosymbiont to its eggs, and the eggs are fertilized already with the right symbiont. So that's why there is a lot of specificity. In other cases, the gametes are not infected with the symbiont, but the mucus surrounded the gamete bundle contains symbionts. So they are seeded also with the parent colony's specific algal species fairly quickly.
In a given cnidaria, they experience cell death and cell turnover, right?
Yeah, and there is the host controls algal cell division inside its cells. There is control of the mitotic division inside a host cell. So it's not like they proliferate uncontrollably. There is regulation of the algal cell cycle, the cell division by the host.
Well, what percentage of cells, I don't even know if you can tell, but what percentage of constituent cells take up the dinoflagellate? And then when one of those dies, I guess they'd all be filled with the dinoflagellates, or is there a turnover?
Yeah, they can shed. They can exocytose and they can shed. There is evidence of that, and there's reviews on this and how they can control the, like in bleaching, if there is a mild thermal stress, there is things that the algae can do in terms of controlling the interaction. They can control the pigment and how much pigment they make, whereas the host can control how many cells it harbors. So there is, even throughout the year, there is variation in an animal of how much chlorophyll there is inside its tissues and how many algal cells there are. If there is thermal stress, then they start to adjust, both of them try to coordinate these adjustments so that they can continue to work effectively until they reach a point where the physiological demands of each are not any longer in sync, and then they have to split up. So the algae decides to leave, or the host decides to expel it, and then sometimes if it's too dramatic, there is actually tissue sloughing and tissue necrosis so that they can get rid of big portions of the colony to get rid of the algal cells. So there is all different kind of physiological processes take place, not only cellular, to address to keep a balance or to control the abundance of algal cells within the host tissue.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Has anyone looked at the the dinoflagellates themselves in the free living stage and looked at their genes and their metabolome, and then looked at them once they're captive inside of a host cell and looked for the differences. And same with the host cell, looking at the gene expression changes with the endosymbiosis of the dinoflagellate or not.
Yeah, I think, we as a community first started looking at gene expression, but now there is more multi-omic approaches, especially metabolomic approaches, to look at this. They're kind of in their infancy, but more and more groups are introducing these. It's hard to work with coral larvae because you only get them usually once a year. So there are people trying to develop other model systems. Like in my lab, we study these jellyfish because we can infect it whenever we want with whatever symbionts we want. People are also looking at anemones that can live with and without symbionts, and then look at the processes of acquisition and breakdown of the symbiosis in a more controlled lab setting, which you can't necessarily do with coral larvae because you only get one shot a year, and that really restricts what you can do. So yeah, people are definitely examining this interaction from an omics perspective, more in depth.
Okay. Well, what do you think is going to be important about figuring out the relationship between these endosymbionts and the cnidaria? Like, what are you looking to look at the effects of climate change and how to maybe repatriate these dinoflagellates in corals, or what's your aim?
Well, there's a lot of interest in the scientific community to try to find traits that confer resilience to corals. We're far away from the reef, so it's hard for us to do that, right? But we're trying to use a more kind of basic science approach to understand this, and that's why we switched to these. We haven't completely switched, but we are developing this jellyfish model because we're interested in how the symbiosis impacts development. Because in corals, we know that when the coral larvae acquire the photosymbiont, there is a lot of developmental processes taking place. They go from a swimming stage to a sessile stage. They become the calcifiers that start to deposit skeleton, and that is all linked to the symbiosis. So by trying to have a model system like a lab rat, we're hoping to be able to zoom into processes that we can't in the lab and try to understand also the breakdown of the symbiosis, which we can do in the jelly. So I guess our contribution to that is in developing this model system and understanding better things that can be addressed with coral larvae. And I guess I can tell you a little bit more about the microbiome stuff. Microbes have been linked to potentially bleaching, to disease, to different stressors, anthropogenic stressors. So we've gone into looking at microbiome composition across different reefs, across different species, and across developmental stages, and see how microbial communities are being impacted by all these different physiological, developmental, and biogeographic conditions. And that's more of a kind of coral experimental field-based approach that is also providing insight into whether there is microbial symbionts that are also as important as the algae. And the microbial community is a lot more complex and less studied than the algal photosymbionts, so it's going to take time, but we are getting some interesting observations from that.
Okay. Have you observed, bleached coral like in situ? Have you gone and been able to sample it and look at it structurally and, to gain some insight on, what a healthy coral looks like versus a bleached one, like in person?
Yeah. I've seen it in the fields, when I had scheduled field trips to collect for actually coral spawning in the Caribbean, which is where I've done most of my work. takes place at the end of summer because it's linked to lunar cycles and temperature. So it's warmer, and that's when they do it. So that's when it gets hotter, and some years it's been pretty bad. So I was in Panama and Florida for the 2005 bleaching event. That was pretty bad. We didn't sample to look at things structurally, but let me think. One year we were looking at biomineralization in corals. This is a different project, and I had a student looking at the skeletal matrix of samples that we collected from a coral hatchery and from field expeditions. And we had some bleach samples and we had some sick samples. And he looked at the skeleton, not at the tissue, which is what most people tend to look at because that's where the live organism is. And we found major differences even in the skeletal matrix, which is that's why I remember this, because you asked about structural changes. So I don't know if there are structural changes that are visible, but there are definitely some structural changes because the proteins that make up the skeletal matrix were clearly different in these samples that were sick and bleached.
Interesting.
I think it would be worthwhile looking at ultrastructural differences. With the naked eye, I don't think you can see anything structurally that you can tell between a bleached and a normal coral, a healthy coral. All you see is the paling of the tissue, which can be transient, right? If it's not too severe, the algae will grow back up and then the tissue will look normal again. But if it's persistent, then maybe something is affecting, and I suspect it is because the coral doesn't have the energy to deposit the skeleton that it normally does. So it must be making less skeleton, but these are temporal scales that are relatively short, right? So you need to be able to look at ultrastructure, I would think, not something that you can look at with the naked eye or even with a regular microscope, if that makes sense.
Yeah, okay. What about, are you looking at any of the phages that would affect, the dinoflagellates and affect the coral or the cnidaria, and then, what happens to them once there's an endosymbiosis?
No, but there are people actively looking at that, and I can give you names of people to interview because that's really interesting. There didn't used to be, but there are more people looking at that now. So I can't tell you very much.
It's okay. No, I know there's levels upon levels of complexity, and no one scientist can even begin to look at it all. So that's why I figured I'd ask.
Yeah.
Well, very good. Yeah.
Okay.
Monica, what's the best way for people to find out more about your work?
They can go to my website or look me up on Twitter. My lab website is medinalab.org, and my Twitter handle is MO Medina Munoz.
Okay. Well, very good. Well, Monica, thanks for coming on. I really appreciate it.
Okay. Thank you for the invitation.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.